Hello, everybody. Obviously, we'll be doing this next little section of the course on remote learning, which most of you should be familiar with me so far. But if not, basically, I will record a video for each lesson, and then there will be a quiz that goes along with that, and I'll tell you during the video when to pause and answer the questions in the quiz. Obviously, some of this information can be, a bit harder to understand if you don't get to ask questions. So please make sure you let me know if there are any questions, or we can always jump on to Zoom to go through. What we're going to look at today is asexual reproduction. We'll move on to looking at sexual reproduction next, but first we're going to look at that. So we're just going to have a look at which types of organisms reproduce asexually. Look at some different types of asexual reproduction. We'll touch on the advantages and disadvantages of asexual reproduction. And also we're going to look at cloning, which is obviously a pretty cool thing to look at. So asexual reproduction basically is when living things are able to arise from only one parent. Because of that, because there's only one parent, there's only one source of hereditary information. And by that we mean DNA. So I know we haven't looked at sexual reproduction properly yet, but obviously we have looked at things like fertilization, and you're pretty familiar with how it all occurs. But with sexual reproduction, we have gametes, so we have a sperm and an egg fuse and fertilize. But with asexual reproduction, that doesn't happen because there's only one parent. So there's only one source of DNA. Because of that, the organisms, also the offspring that are born, are genetically identical to the parent, unless mutation occurs, which is like a random change to DNA. Remember, mutation happens randomly, it can't be planned. So if that doesn't happen, which usually it doesn't, the offspring, let's say all of these guys down here, will have been exact copies of their parents, which are exact copies of that parent, which were exact copies of this parent at the top there. Okay, so just make sure you've jotted down those notes, or I've included the slides if you want to use them, and then jump into the quiz now, pause this and answer question 1. Okay, we're going to look at 4 different examples of asexual reproduction. Okay, so and there's a slide for each one of these. So we'll look at fission, which many of you will already be familiar with, budding, vegetative propagation, nice big fancy name, and then also spore formation. So we'll start with fission. Okay, another term or a particular example of this is binary fission, which if you're doing biomedical science, you should be very familiar with this, which is the way that bacteria reproduce. We also see it in amoebas, which are single-celled eukaryote organisms. This involves the parent cell, so the bacteria or the amoeba, dividing to create 2 daughter cells, similar to mitosis. We call them daughter cells. Because of the way this occurs, and it can happen relatively quickly, like this can, in some organisms, only take about 10 minutes. So some bacteria can divide very, very quickly. And it has an exponential growth rate. So by that we mean we go from 1 to 2. Then if both of those 2 divide, we have 4. If all 4 of those divide, we have 8, then 16. So it grows very quickly. If we were to graph the growth of or the reproduction by fission, it would look something like that. Okay, very rapid growth. And this is why, if you get a bacterial infection, you can get sick quite quickly because those bacteria are just dividing very, very rapidly. The steps of binary fission, it's very simple in comparison to mitosis. So first, the DNA duplicates, which is kind of what's happening up here. This is the DNA inside a bacteria, and then the cell elongates. Sorry, these pictures are supposed to show up. I'll put them up now. So the DNA duplicates, so we get 2 copies here, exact copies, and the cell has actually elongated, it's stretched out a little bit there. Then we get a thing called the septum forming. Okay, so the septum is basically just this bridge in the middle, same way as the inner part of the nose is called the septum, or you might remember from the heart, that middle bit is called the septum. You get a thing called the septum forming here, which is going to separate those 2 cells, and then the cell splits into 2. Okay, so quite simply, it's just the DNA doubling, the cell getting bigger, and then splitting into 2 identical copies. Next, we'll look at budding. So budding is a type of reproduction that occurs in flatworms and also in yeast. So if any of you are bakers, you've probably used yeast before. We use it in bread. It's used in making alcohol. It's used in making pizza doughs, all of those kind of things. And yeast are just these kind of small organisms like this. In this type of reproduction, the cell doesn't kind of split into 2 exact copies straight away. What actually happens is we get an outward growth, which is what is called a bud, so that's what's called budding, starts to develop from the organism. So you can see that here. It's not an exact copy of this yet. It's basically starting to grow out from there. It then will grow into full size and end up being kind of an exact replica of the original. But there's 2 things that can happen when that bud grows. The first thing is that it will detach like that. So the bud detaches and then it will grow to its full size. Or quite often what happens is those buds will stay attached to the parent and then they'll create their own buds and we end up getting colonies. So a colony is just a big group. We talk about bacteria forming colonies, but also yeast will form colonies if the buds stay attached. Also, there are questions on this, but it's going to be after we've looked at all 4 because you're going to be comparing the 4 different types. Another type is called vegetative propagation. Okay, this only happens in plants and it's usually something that's done artificially. And when we look through the process, you'll understand why. But basically, part of a plant is detached. Like here, we've got this stem here has been cut. It's usually cut on an angle just to help it grow again. So this bit gets removed and then it grows into a new plant if it's put into the right conditions. So you can see here, if you were to cut a bit of a plant and usually you'd put it in water first and allow the roots to start forming, and then you can plant it in soil and it will grow to be a new plant. It may not look identical to the parent because obviously the leaves might grow in a different direction, but genetically it is identical because it has not been cross-pollinated with another plant. Okay, all of the genetic information came from this original plant. And you can see obviously that the reason this generally happens artificially is that it's quite unlikely that a plant will get cut on a certain angle and then be put into water, which will allow its roots to grow, and then it will be put into a nice soil condition. That doesn't generally happen naturally, but technically it could if those conditions were met. This is used pretty frequently in gardening and in farming. Some of you might have done this at home, or you might have parents that you see take clippings from plants and regrow them somewhere else. And the last one is called spore formation. This is a pretty cool type of reproduction. It occurs in fungi, so all types of mushrooms, in protists, which are very, very small organisms, and some plants do this as well. So mosses and ferns will do this, and this tends to happen more naturally. So these organisms form things called spores, and a spore is basically a single cell filled with DNA. Okay, so it's basically like having, I guess, like having little zygotes, because it has all the genetic information. Remember, it doesn't need to be fertilized. That then are able to form new organisms. So if you look at this picture here, this shows the underside of a type of fern. If you're ever out in places like the Dandenongs, it's full of these ferns. I really like those kind of wet rainforesty conditions. If you look at the bottom part of a fern leaf, you'll see all these little dots, and they are spores. They're still attached to the plant at this stage, but then what will happen is the spores get released from the organism, and then they will go and settle somewhere else. So that they'll drop to the ground and settle on the soil. And then when the right conditions are there, so the right amount of light, the right water, and the right temperature, the new organism will grow. It's pretty cool, and it's obviously those spores, once they're released, get picked up by wind. So you can see here this picture of mushrooms releasing spores. Those spores will spread all around the area. So sometimes you might have wondered how, you know, a plant that's in one location could give rise to plants, you know, in other locations. That's because of these spores. And you see this every year with mushrooms. Generally, if you see like one cluster of mushrooms, there'll be loads more around it because those spores have settled in those locations. Okay, so that's the 4 types of asexual reproduction. Like I said, I want you to jump into the quiz and have a go at question 2. Basically, there's 8 different statements there, and for each one, I want you to say whether that is relating to fission, to budding, to vegetative propagation, or to spore formation, and then come back to the video once you are done. As with, you know, most things in biology, there are advantages and disadvantages to asexual reproduction. The big advantage for organisms that are able to reproduce asexually is because they don't need a mate, it conserves energy. So, you know, a lot of organisms that need to mate with something else to reproduce will waste a lot of energy trying to find a mate, going through all the processes of mating. It doesn't require that. Also, if there's no mate available, the species won't die out. So that makes it a selective advantage for a species. It's usually very, very fast, so can produce many offspring quite quickly. If you think back to the binary fission and that exponential reproduction rate, that can happen very, very quickly. Same thing with yeast, with the budding, they can produce multiple buds quite quickly. Another advantage is because all of the offspring are genetically identical, if the parent was well suited to their environment, then the offspring will be as well. Okay, so if the parent is surviving really well in a particular environment, all of the offspring are going to survive well in that environment. The disadvantages of this, the first one is that there's no genetic variation. So we've said that that's an advantage down here if they suit their environment, but it can be a problem, firstly, if they don't suit their environment, or more likely, if the environment changes. Okay, so let's say there was a group of bacteria in a, you know, very cold environment and they were suited really, really well to that, and then that environment changed, it got warmer, and now the bacteria aren't suited to it. That population would die out. If you had genetic variation, there might be some individuals that are suited to it, and those ones would survive, kind of that natural selection process. Okay, so that's a disadvantage here. Another disadvantage is that because you can produce lots of offspring very quickly, you can end up with large numbers of offspring that have to compete for resources. If we're thinking of things like bacteria or yeast, resources are going to be things like space. Is there enough space for them to divide? Is there enough oxygen available? Are there nutrients available? So, you know, if they're not able to access those things, then certain individuals will die out. Okay, so just want you to jump into question 3 and just write a short paragraph kind of outlining advantages and disadvantages of asexual reproduction. All right. We're going to have a look at cloning. Cloning is a pretty cool thing that can happen. There is a lot of ethical debate around it, as you can imagine. It can be a relatively complex process. So if you watch this and you don't quite understand the science side of it, that's okay. But yeah, you might be able to follow this really well. But don't stress too much if it's a little bit confusing. Cloning is basically the artificial process of making an identical copy. Okay, this can be done either to entire organisms, so cloning, an entire sheep, for example, or it can involve cloning genes, so bits of DNA, and this happens very, very commonly for scientific research. Okay, there's not a huge amount of ethical dilemma with cloning genes because a little section of DNA is not a person, is not an individual, but obviously it can lead to other things. We're going to look at two different methods of cloning, so two different ways that we can achieve making identical copies. The first one is called embryo splitting, and the second one is called nuclear transfer. So we'll go through both of these now. Like I said, don't stress too much if it's a little bit confusing or you can't fully get your head around how these processes occur. So embryo splitting is the first one we're going to look at. If we look at the term, kind of makes sense. Basically, we're going to get an embryo, we're going to split it into two. What happens here is that an egg is removed from a donor female, and that egg gets fertilized. Okay, so we've got an egg from, we're going to use the example here of the sheep, egg from the sheep. Now, I can't actually remember the chromosome number of sheep, but I'm just going to put 23, which is humans. I think it is different for sheep, but let's say there's 23 chromosomes in that egg, and then it's fertilized in a lab by a sperm that has 23 chromosomes, so we end up with a zygote. Okay, let's just say that. So the same that would happen, it's just happening artificially. So that zygote is going to start separating, just as it would in normal kind of fertilization. So it'll divide by mitosis and form a two-cell stage. In embryo splitting, those cells get separated. Okay, so as soon as the zygote has separated into two cells, those two cells get put into different petri dishes, basically, different things in a lab, and then they're left to divide individually. Okay, so each of those two cells will then divide into two different blastocysts. Okay, so mitosis is occurring in both of them. They're both identical. Both of those blastocysts will be identical to the genetic information in that zygote. The two blastocysts are then implanted. This can happen in one surrogate mother. So you're basically putting in two eggs, basically as two embryos, which is pretty much what happens with twins. Or they could actually be put into two different surrogate mothers. Okay, so the surrogate mother is basically just a carrier. Okay, the offspring will not have any of the genetic information of the surrogate. All the genetic information came from the donor at the beginning. Okay, so the surrogate is just going to carry the embryos through gestation, so through that pregnancy period, and then eventually give birth to the offspring. Okay, so in this example, we get offspring that are genetically identical to each other. So you can see down here, these adorable little sheepies, identical to each other, but they're not identical to the donor egg. In the same way that identical twins are not identical to their mother, they're not identical to their father, but they are both identical to each other because they share the same DNA from the mother and the father. Okay. If you want, you could go into question 4. Again, it is a comparison. You could go through and click all the things so far that apply to embryo splitting. and then you'll be able to go back and finish it when we look at the other example. But in a very short way, embryo splitting is basically just like producing identical twins, but it's done artificially. Right, nuclear transfer is slightly more complex. And when we say nuclear, we are referring to the nucleus. Okay, so in this, I'll basically say what the outcome is. The outcome is going to be an offspring, which is, in this example, a cow that is genetically identical to a donor. Okay, so it's not, I mean, technically, obviously, the cow will be born as a baby and it will grow up, but genetically, it is identical to its donor parent. So in this, a body cell, and a body cell being a cell that has all 46 chromosomes, again, that's the human number, I'm not sure what it is for cows, but it's not an egg, it's not a sperm, it's an actual body cell, is removed from one animal, and that one is the donor. Okay, so this here is our donor. We take a body cells from it. An egg then gets removed from a different cow. Okay, so this is the egg cow over here. Okay, remember the egg only has 23 chromosomes. It has half the number of whatever the diploid number is for that organism. Okay, the nucleus of the egg gets removed. Okay, so if this was the egg, and it's the nucleus here with 23 chromosomes in it, and then we've got our donor cell with 46 chromosomes, because that's a full cell, the nucleus from the egg gets taken out. Okay, because that's only half of an offspring. The donor nucleus is then inserted into that egg. Okay, so the donor nucleus with its diploid or its pairs of chromosomes are inserted into that hollow egg. Now, technically, I guess we could say this is now basically a zygote, because that entire organism is going to stem from this egg. It's just that the egg was never fertilized, it was just given a whole set of DNA. That egg with the donor DNA is now implanted into a surrogate and gestation occurs. Okay, so we've basically got 3 cows involved in this process. We've got the donor cow, we've got the egg donor, who literally is just giving a hollow egg. We're not using any of its DNA. And then we've got the surrogate, which is actually going to carry and develop that cow during its gestation, during its pregnancy period, and then give birth to it. And then we'll end up with our offspring, which is genetically identical to this cow up here. Okay, it doesn't have any DNA from this cow. It doesn't have any DNA from this cow. All of its DNA came from the body cell taken from that first cow. Okay, it's a pretty cool process when you think about it. But as I said, this can be quite confusing stuff to learn through a slide. So if you don't fully get it, that's completely fine. Have a go at answering question 4. Some of the statements apply just to embryo splitting, some apply just to nuclear transfer, and some apply to both. Okay, I'm not going to go over too much how cloning is used in agriculture, but it is used in growing crops. This is looking at some coconut crops that are all genetically identical. They've been cloned, and then they still have to be planted and grow like everything else. I think that's a really common misconception people have with cloning, is that they think, clones are produced, in some weird kind of sack in a lab. That's not true. It's just the actual, the genetic information is cloned, and then it still has to be planted or go into a uterus and develop as normal. So it happens quite often in crops and in plants, and also it has happened a bit with animals. So Dolly the sheep was the first animal to be cloned. But, you know, we see it often with things like livestock might be cloned because they have, you know, a really great color that can be used for leather, or they produce really, really good milk, things like that. What I want you to do is just have a think about asexual reproduction. You've started looking at ethics a little bit. I want you to go in to question 5 and just, you know, this is really just your suggestions. What are some good things about cloning? How could it be used? How can it benefit people and farmers? And what are some potential risks? And if you want to get all sci-fi and conspiracy on me, do it. I love that stuff. You know, if you think this is going to lead to a super breed of humans and you think that's a potential issue, then absolutely feel free to write all about it. And that is us done for today. Thank you very much.